PDF, sir. PDF. Hello students, good evening. Can you all hear me? Hello.
Good evening.
Yeah. So only Purna is here. Others are yet to come, right?
Yes.
Yeah. So we'll wait for a few more minutes and then we will start the class. Okay.
Okay.
And Purna, can you see my screen? The
Yes,
screen
sir.
is visible. Fine, right? So can you all able to see the screen? Hello, students. Hello, can you all able to see the screen?
Yes, sir.
Okay. Okay. And so today we are going to discuss about the heredity and evolution. And so primarily focusing on the concepts of heredity. Fine. So now, like we discussed earlier, like what evolution means. Hello, students, what evolution means? Remember? How can you define evolution? Evolution is what? Three important things. Origin of evolution. Forget the definition for evolution. Evolution is what? Hello? If all are silent, I feel like I'm not audible to you. How can you define evolution?
Sir, we can say that, sir,
Yes, yes. And if you can't define evolution, and then no use of studying heredity. Okay, so fine. So listen, everybody just like that should give me definition for evolutions. Fine, good. So evolution is the origin of life form, and three important things we saw in evolution. Keep in mind how we saw a lot of theories also in person, like so it is the story of origin of what? Earth. Okay, do you remember origin of earth? Origin of first life form, and then diverse form of life. Okay, three important things. Evolution talks about the origin of earth, first life form, and from that first life form, how different group of organisms, different kind of organisms are evolved. That is evolution, the origin, the formation. Okay, the reason for asking the question of evolution is there are three important components of evolution. What are the three important components of evolution? So what are the three important components of evolution?
don't know, sir.
Don't know. Very good. Okay. The three important components of evolution are heredity. Okay. Variation. Natural selection. Okay. So the components of evolutions. What? Evolution comprises of three important components. What are they? Heredity, variation, natural selection. Okay, heredity, variation, natural selection. What is heredity? How can you define heredity?
Transfer of traits.
Yeah, like parents produces progeny of their own type by reproduction. So here the transfer of characters from parents to progeny in the form of DNA. Okay, the parent transfers DNA to the progeny. Okay, that process by reproduction. So that concept of evolution is known as heredity. Okay, parent transfers the characters to the progeny, all of their characters to the progeny. Okay, that process, inheritance of traits from parents to progeny, that is heredity. Okay, and here next variation is the degree in which, okay, the degree in which progeny differs from the parents. Okay, the degree in which the progeny differs from the parent is called variation. Okay, and I think in the class we used a kind of symbols. The same symbol I would like to give you all for your reference. Again, just to remind things like, so this is the organism. You can say, remember the example, organism is round and this one form. So the parent produces progeny of its own type. So this process is what? Inheritance. Okay, inheritance. The progeny, they are the progeny or identical to the parents. Progeny or identical to the parents. That is inheritance. Variation in which the progeny differs from the parents. You can see here one organism is like inherited. Similarly, this is similar to the parents. And here you can see dissimilarities to the parents and this progeny. So this is the results of what? Variation. Okay, the progeny, look at that here. It is circle and one single just to represent its form. And here you can see the form itself is slowly changed. This is called variation. Okay, how the progeny are similar to the parent, that is by inheritance or heredity. Variation in the sense how the progeny, in what degree it differs from the parents. That is what the variation. Now you have here one old species and new species. So this is the old one and this is the new one. Okay, old and new. Which species will have more survival advantage? Do you remember? I gave you 4 options. First, old will have more survival advantage, new will not have. Or ulta, old do not have more survival advantage, but new will have. And then third, both will have equal survival advantage. Fourth option is both will not have the survival advantage. This is determined by,
Natural selection.
yeah, the nature only determines. That's why this is called what? Natural selection. So evolution is the origin of earth, life form, and diverse form of life. It has 3 components. So all the processes of evolutions are achieved by these 3 processes. So they are the major components of evolutions. They are called heredity, variation, and natural selections. Heredity includes like the inheritance pattern. Heredity is the studying of the inheritance pattern. What is inheritance pattern? How characters are transferred from parents to progeny. That is inheritance. And then variation in the sense, like in inheritance, the progeny are similar to the parents because of the similarities in the DNA. In variation, because of internal or external factors, mutations or whatever, the progeny differs from the parents. So maybe new accumulation of variation, results in speciations. little variation, and finally results in speciation. Thus, from one life, different life forms evolved. And then here one more, to understand the important component of evolution, we used this example who will have more survival advantage. So that should be determined by, supported by the nature. So hence that is called what? The natural selection. Hope you know what is evolution, what is variation, what is inheritance or natural selection. So the same picture. So this picture also, the parent produces progeny and then they insist on the variations. Okay, how maybe the color or any other characters, how they differ from each other. So which insist on the variation and inheritance. Okay, fine, good. So that's the very important thing. Okay, so please understand those terms. Okay, evolution, heredity, variation, natural selection, all these things. Okay, and you know that the processes of reproduction, especially like all type of reproduction, it may be sexual or asexual, ensures these all processes. Okay, reproduction plays a crucial role in all these processes. Okay, fine. This is what all the questions which we are discussing. Do all the variation in a species chances surviving the environment in which they themselves? It depends upon nature. Okay, nature only decides what need to be survived. Okay, fine. Now, so heredity, as we know, the science of inheritance is called what? Heredity. Okay, they are explaining about some characters here. Okay, how characters, heredity is the science of inheritance of triads. Okay, the technical term used to define the heredity is triads. Okay, now they are giving as some examples. Okay, so what this diagram represents? What this diagram represents?
So variation.
Okay, variation. Very good. What are the two type of variations here?
So the lobe is attached to the skin and the other one is free.
Yeah, fine. Very good. So this is actually called, we call it as a term called what? Okay, the character. It is a phenotype. What is phenotype?
Physical character.
Yeah, like the which you can see. Okay, and physical trait. Very good. So here two traits are given. Free lobes and attached lobes. Okay, this is one example of trait. And now my question is, which is having more survival advantage? Which trait? Will be there any? See, actually, some traits, okay, some traits, physical traits, actually do not have much of the effects. In no way it is going to affect your life. Okay, directly it is not going to play a crucial role. Okay, some traits do not affect the life of the individuals. Some phenotypic traits. I will ask you some other example, give you some other example. Like your blood group. Okay, some are positive, maybe A, B, O blood group. Okay, A, B, O blood grouping. Some will have negative blood group. Will it be having any effects on your life, impacts on your life? Blood group is it right? Right? It is determined by the genes which you get from your parents. Is it going to affect your life or Is there any significance of having a particular blood group? You get my question? Hello, students, you get my question?
Can you repeat the question, sir?
See, phenotype of air. You can see the first picture shows free air lobe. The second picture shows attached air lobe. Is it going to affect your life whether you have free or attached air lobe? No. My question is no. Second example I gave you, somebody will have A positive. Somebody will have A negative. Again, that's a trait, character. We inherited from our parents. Will this type of blood group, a particular blood group, phenotype, character, is going to affect your life, impact your life in any way?
No.
No, blood group type. See, lobe is not going to affect. I'm sure this is not going to affect any of your life process. But the kind of blood group, will it affect your life?
I guess
Impact
yes.
yes. How?
So
How?
different blood groups have different properties.
Okay. Then
And availability of blood.
yeah, maybe availability. Very good. So listen, we vary in blood groups, vary in triads, and getting blood donors. Okay, if the wild type is positive and mutant type is negative, the dominant is positive and recessive is negative, and definitely the kind of blood group in emergency will affect your life. So what I want to say is some variation, some phenotypic variation will not have any impact on your life, but some variation will definitely have impacts on life. For example, one more clear example I can give you. If you are having hemophilia gene, you will get hemophilia. If you do not have hemophilia gene, you don't get hemophilia. So some disease gene, if it is there in your genome, you get. If it is not there, you do not. So the characters which we inherit varies from one person to another person. It will have lot of impacts. Sometimes it may not have impacts also. You understand my point? Hello?
Yes, sir. Yes, sir.
Okay. Now, rules of inheritance. Say what are the different mechanisms by which the characters are going to be inherited. So this is what we are going to discuss now. So let me tell you some important things. So please listen to that, and then I will start asking you the questions. Okay, fine. We call Graham Mendel as the father of genetics because his hybridization experiments on pea plants helped us to understand the rules of inheritance. Keep the point. Graham Mendel's hybridization experiments on pea plants helped us to understand the rules of inheritance. That's the important thing. Okay, and then what next we should know is the characters which are fascinating and contrasting. Okay, the characters selected by Mendel. So that you should know. Okay, the kind of contrasting characters selected by Mendel on pea plants. Do you all remember the table of the 7 contrasting characters? Okay, it is listed here. You must keep that in mind. All the characters. For example, first, you tell me now, the first one, what is it?
Tall and
What?
short.
Yeah, character. First, tall and off. No, the character is height. Second, idilla
Type of
two. What is dominant? What is recessive? Dominant and recessive traits were there. Which is dominant?
tall,
Which is recessive?
short.
Okay, short or doff. Second character?
Graph.
Hmm? Ah,
Type of seed, shape of seed.
okay, seed shape. Which is dominant?
Round.
Round,
Round.
Recessive?
wrinkle.
Wrinkled.
Very good.
Color.
Okay. Okay, seed color. Which is dominant?
Yellow. Green.
And then next,
Color of flowers.
flower color. Okay. Which is dominant?
White.
Really? White or purple?
Purple.
White, sir.
That's-
Okay. Then
Position of flowers.
here. What is dominant?
No, I don't know, sir.
Axial. And then terminal. Axial and terminal. Okay. Okay. Axial means what? See, like this. In all axis, you have the flower. That's what axial means. Terminal in the sense, only in the terminal end, you see the flowers. That is terminal. Okay, terminal. In all axis, you see. Axial and terminal. So next character. Is next character.
So pot shape.
Okay. Pot shape. Which is dominant. Pot shape. Yes.
I guess full.
Mm. Constricted. Full and constricted. Okay, I cannot write well, but constricted. Here it's not writing. Okay. Constricted. Okay. Full and constricted. Full means like this. Constricted in the sense like this. Okay. Full and constricted. And then
Pot color.
pot color. What is dominant?
Green.
Green. Recessive?
Yellow.
Yellow.
Okay, very good. So you must be thorough with all these characters, right? what is the important character, and then from that, which is the dominant one, what is the recessive one. So that's very, very important. Fine. So now, some simple questions. Okay. What is self-fertilization or self-pollination, cross-pollination?
So self-pollination happens between the same flower in the same flower
Okay.
or the flowers of the same plant.
Okay. Very good. So what is homozygous condition, heterozygous condition?
So homozygous is the same type of allele and heterozygous different alleles.
Yeah. Look at here. Homozygous for dominant. How can you describe this? Homozygous for dominant. And then here, homozygous for recessive. Right? Both are homozygous. And what is this? Genotype. What is this? What genotype is this? How can you describe this? How can you describe this? This is the genotype. How can you describe? Capital T, small t. How can you describe this?
Homozygous.
Heterozygous.
Heterozygous. Okay, heterozygous conditions. Okay, so you must know about genotype, the phenotype, homozygous condition, heterozygous condition, all these things, right? Fine. So here in Graham Mendel's first experiments, hope you all know how to work out monohybrid and dihybrid cross.
Yes,
Right?
sir.
Yes. Fine. So this is like the tall plant crossed with the dwarf plant. The genotype is this is homozygous for tall, this is homozygous for recessiveness. But in the F1 generation, you can see all the plants are resemble just one plant, the dominant character. Okay, first. And then he crossed the F1 plants. Okay, just the F1 plants, all the F1 plants were heterozygous. So they when they crossed the F1 plants, like he can see tall versus even dwarf, both of the character. So this is Graham Mendel's monohybrid cross experiments. Okay, he selected one homozygous tall, homozygous recessive. In F1, all were tall, and then he crossed F1 progeny, and he got in the second F2 generation, he got tall as well as the dwarf character. So this did not appear in F1 generation, appeared only in F2 generation, the dwarfness. Okay, this is his experiment. So what is his conclusion? What is the conclusion of this? What's the conclusion of this?
Of the second, this one you're asking, sir.
But based on the monohybrid cross,
That the genes don't get mixed.
very good. First, very important thing, genes do not blend. They stay as such. Tall genes remains as tall, dwarf gene remains as dwarf. Very good. Then
And then law of dominant traits and recessive traits.
very good. Law of dominance and law of segregation. Okay, law of dominance and law of segregation. What the law of dominant insists, what it says?
And it says dominant trait can be exposed in heterozygous and homozygous condition, whereas recessive trait it will be exposed only in homozygous condition.
Okay. Okay. Fine. See, understand this proposal correct. Mendel's proposal. Two copies of factors. Now we call genes controlling traits, okay, means every character is determined by two factors. Okay, we get from the parents, and these two factors, genes, may be identical or may be different. Okay, may be identical in the sense like this, different in the sense like this. This is his first assumption. And then among those two, okay, among those two, the character which is expressed in the F1 generation, he called it as dominant. Okay, he called it as what? Dominant character. And then the character which, okay, the character which he saw in the F2 generation, he called it as recessive character. Can you understand? Based on his experiment, this is his conclusion, like factors we inherit from parents, two factors we inherit, one from Appa and one from Amma. The two may be same or may be different. Two may be same dominant, or two may be same recessive, or maybe one dominant, one recessive. And then the one which character which expressed in the F1 generation, he called it as dominant. The second one, he called it as recessive. Okay, fine, good. So this is law of dominance. And also the law of segregation, what it tells, you can see, even though the dominant characters are expressed in large amount, the recessive trait has equal chances of inheritance. Okay, if it is recessive, it doesn't mean that it will be inherited in less frequency. No, maybe dominant or recessive, they show equal pattern of or similar pattern of inheritance. They involve in meiosis, they get segregated, they get into gametes and inherited to the next generation. So that is actually called what? Law of segregation. Okay, so from monohybrid cross, law of dominance and law of segregation. Now one more question. What is the genotypic ratio and phenotypic ratio in monohybrid cross? That you have to remember. What is the genotypic ratio? Yeah, in monohybrid cross, it is 1:2:1. In the phenotypic ratio is 3:1. Okay, so this 1:1 means 1:2:1. That only 1:2:1. So this is what 1:2:1. And here phenotypic in the sense, this is also tall, also tall. So 1 plus 2 equal to 3 tall, and then 1 dwarf. Okay. Adanala, it is called what? 3:1 ratio. You know the basic understanding of that. Okay, correct. So this is what we should remember in monohybrid cross. Now, Gregor Mendel worked the same kind of experiments in all of the contrasting traits. We saw different contrasting traits. See, he worked for all of these contractors. We just worked now, saw the example of one. But he worked on all of the characters. All of them showed the same pattern of inheritance. Okay, means the almost the 3:1 ratio he could able to get. So that's the main important discovery through monohybrid cross. Okay, in all example, this is another example. Okay, the color purple and white flower. Okay, the dominant purple, recessive white flower. Right. And then now, what is the second experiment?
Dihybrid cross.
Dihybrid cross. Dihybrid cross explains The pattern of inheritance of hmm? Two characters. How two parental characters are inherited. So that's the main thing. Okay, dihybrid cross. Look at that. What example he has taken?
Yellow, green,
Yeah,
round yellow, wrinkled green.
Yeah. Seed shape, right? Seed shape. What is round? Color yellow and green. Correct? Round or wrinkled, yellow or green. Okay. So two parental characters he selected. Right? Right? Okay. Fine. See, listen. He selected two characters, crossed, and then these are the gamete combinations. So this one R here, one Y here, and this one R, one Y here. So the conditions basically he got, and he selfed these, and then he got this heterozygous condition. Hope you can work out this. And then from here, the type of gametes in this heterozygous condition, how these gametes are formed. So this is one thing, okay, here, and then the capital or small y here, and then the small r, capital Y, small r, small y. So like that, different type of gametes can be formed. He just selected two parents like that and then crossed. Okay, so this is the phenotype as well as genotype is explained in this table or square. You can call it as what? What square it is? What square it is?
Punnett square.
Punnett square. Okay, fine. Now you know. So the finally the phenotypic ratio is given like yellow round, the dominant traits, both dominants 9, and then yellow means round green, wrinkled yellow, wrinkled green. In the sense, both the parental characters are present. Okay, in the phenotype you can see the parental character. Like, what are the parental character? What are the parental character? This is one thing, right? This is one thing, one parental character. What is it? What character it is? Green in color, round in shape, one parental character. Is it seen in the progeny? Round green. Yes, it is seen or not?
Yes, sir.
Yes. What is the other parental character?
Wrinkled yellow.
Wrinkled yellow. Is it there in the progeny?
Yes, sir.
Look at that, wrinkled yellow. So, you can see the parental character. Also, what you can see? What you can see? The shuffling of parental character. Here, the dominant characters together. Here, the recessive characters together. Can you understand? So, in the phenotype of the progeny, okay, in the phenotype of the progeny, you can see all, you can see the dominant as well as recessive characters, and then the parental and non-parental characters together. Is it clear? The parental characters as such present, the parental characters shuffled and present. The dominant traits together present, the recessive traits together present. Is it clear? What is the conclusion? What is the impact? What is the rule we need to understand from this dihybrid cross? what we have to understand from this diagram cross?
Independent assortment.
Yeah, means the law of independent assortment. The parental characters may be inherited together, may not be inherited together. Means the inheritance of parental characters are independent of each other. Okay, so that is the main thing. Okay, the parental characters are inherited independently. Okay, independently inherited together or with shuffling. Okay, so that is the main thing. Independent assortment. Okay, independently. That's why independent assortment. Correct? Okay. Hello.
Yes, sir.
Okay.
Yes. Now, some process actually they call it as what? Gene expression. Okay, what is called gene? How characters? See, this question is how do these types get expressed? Means this is the question of in simple way. How can you ask the question is like what is gene expression? Okay, gene expression. What is this process? Gene expression. This is the answer for that question. Means how characters are inherited. Okay, what is the question we saw? How characters are inherited. That's the question. Okay, look at that. How does the mechanism of heredity works? The answer is gene expression. What is called gene expression? DNA is there. The double stranded DNA is there. Okay, the DNA, the gene, it contains the gene. Okay, this is the DNA. From DNA, the genes are present. Okay, from DNA, the genes are present. Genes are transcribed into RNA, or in simple, they call it as mRNA. RNA. Okay, keep in mind just RNA. RNA is again translated into protein. Okay, RNA is translated into protein, and this protein determines the character. Okay, listen carefully. How the mechanism of heredity works? That's the question. The process, the answer process is called by gene expression. What is gene expression? DNA contains gene. Genes are, this process is called what? Transcription. Genes converted into mRNA or messenger RNA. This RNA is converted into protein. This process, they call it as what? Translation. They call it as what? Translation. So, DNA, gene, and then RNA, the protein. This conversion process, this is called gene expression process. So, that's the answer for this question. That is what is explained there also. Okay. So, cellular DNA is transcribed into RNA, and RNA is translated into protein. Okay, so gene to RNA, RNA to protein basically. Okay, so this is one important process. So gene expression for inheritance of characters, gene expression, and then other processes. For example, it is influenced by many internal and external factors. Okay, the inheritance or expression of characters is influenced by internal and external factors. Okay, internal. What are internal factors? What are external factors? External, the environmental factors. Internal, the hormones or whatever. In plants or animals, whatever. Can you understand? The hormones and the internal conditions as well as the external conditions controls the gene expression process. Internally, genes, hormones, etc. And externally, the gene expression also influenced by the condition where we live. Okay, the kind of lifestyle we follow, that also supports the pattern of inheritance. Can you understand? Hello? So, in simple, Grahor Mandel worked on pea plants, have done many hybridization experiments. So he, based on his experiments, the genetic mechanisms we know are the characters expressed by genes. Genes may be dominant or recessive. And then the genes from parents may be, set of genes from parents may be inherited together, or they get shuffled and then inherited. Okay, so these are all the basic findings, rules of genetics. Okay, fine. Now, the another important thing is where the genes are present. In a DNA. DNA is present in the form of what? Chromosomes. Okay, so you should know what is DNA, what is gene, and then what is chromosome. Okay, so DNA, chromosome, as well as genes. Okay, so basic differences you should understand. How can you define DNA? How can you define DNA?
Functional unit of life.
Yeah, functional unit of life. Just a single long thread of DNA. Okay. What they are telling is here is DNA carries gene. Look at that in human DNA. They may ask one question. DNA is present in a, you know, what is the length of DNA? 2.2 meter. In one cell in humans, the DNA is present in 2.2 meter. Is it present in a continuous form or discontinuous form? In prokaryotes, DNA is present in a continuous form, which is circular. In eukaryotes, like us, plants, animals, humans, and all, DNA is present in discontinuous form. Okay, discontinuous form. The segments, individual segments are called what? Chromosomes. Okay, individual segments. Okay, the independent pieces, the separate pieces are called what? Chromosomes. And then we have paternal chromosome, maternal chromosomes. Paternal means appa, maternal means one from mama. That's why chromosome, we call it as exist in pair. Okay, chromosome exist in pair. That you should know. So paternal, maternal origin. Can you understand? DNA, genes, chromosomes. Okay, all these terms you should know. And difference between all are DNA, mottama DNA genes now, but definition for all these three molecules you should know, right? Okay, fine. Next, sex determination. What are the factors which determine sexes? Okay, what are the factors which determine sexes?
The genes, the chromosomes.
Okay, some rely on environmental factors. Okay, example, reptiles. Okay, in reptiles, if high temperature develop into female, low temperature develop into male. Can you understand? The egg, in what temperature it is incubated? Temperature above 30 degrees Celsius, female, below 30 degrees Celsius, male. So temperature can influence the sex. Okay. And some organisms like snails can change sex in its life cycle. Okay, they may be male at particular time, and then depends upon the conditions, they can change their sex also. Okay, interchangeable sex. Second important thing. Okay, one is temperature determined, and then even sex is interchanged. And finally, what important thing is, we should understand this chromosomes. Okay, chromosomes, sex chromosomes determine sex. The genetic basis means that is the genetic inheritance. Okay, example in humans, you can see if it is XX female, XY male, the kind of sex chromosome we have determines our sex. Okay, and then honeybees. Okay, honeybees. You remember how the sex in honeybee is determined? Honeybees are having 16 chromosomes in its. Listen, 16 chromosomes. Honeybees are having 16 chromosome in its haploid condition. So, if the egg is fertilized, 16 plus 16. So, chromosome number is 32. This will develop into female. If the egg is unfertilized, will develop into male. Okay, so the process of fertilization determines sex. Now you tell me, what are the different factors determine sex? Environment, like temperature, and then even it can be changed. Interchanging of sex depends upon the conditions, and then chromosomes. Third one is chromosomes determining the sex, and then the process of fertilization determining the sex. Okay, so these are all the things which are connected with the factors determining sex of the organism. Okay, so this is about the heredity chapter, important aspect of heredity chapter. Okay, if anything else you cannot follow or any concepts little confusing for you, you can ask me. Any mechanism is not clear. Hello, students?
Yes, sir.
Oh, then what I can take happily? Oh, understood everything?
That's it.
Yes, sir.
Maybe. Okay, fine. So, please go through this chapter well. If you cannot follow anything, you can contact us. And this is very easy, but you have to be carefully understand what kind of questions are asked. So, the terms, like, to understand the questions, you should be clear about the different terms used in genetics or heredity. You can say like genotype, phenotype, dominant, recessive, homozygous condition, heterozygous condition. Okay, filial 1 generation, filial 2 generation. Okay, so all these terms, because in questions sometimes they do not mention the genotype, they just talk on phenotype. Okay, sometimes they are talking about heterozygous for tall. You can understand, you can know what is the genotype. Okay, or if they are giving you only the genotype, you should know how to describe the phenotype. Okay, so genotype, phenotype, that basic understanding is very, very important. and the different kind of crosses, monohybrid, dihybrid crosses. Okay, fine. So that's the most important thing in this particular chapter, the terms. Okay, if you understand the terms, you understand the mechanisms. Okay, fine. So all the best for your test. Take care. Thank you. Hello. Ma'am, the எனக்கு எப்போ மேம் கிளாஸ்?